Hello, everyone, and welcome to our fourth episode of the Neural Network. My name is Vasudevan Lakshmin, and I'm here with my co-host,
Armaan and I. Today we'll be joined by Joseph Yu, another senior at Mass Academy, to discuss his previous experience and projects, along with other AI things. One of the main projects we'll be discussing today is the disease detection model he developed based on immune footprints. Joseph, would you like to introduce yourself first?
Yeah, sure. Thanks for having me. So I'm Joseph. Like you said, I'm a senior, and I guess I'm someone who really likes doing machine learning and learning more about it. And as you mentioned, my STEM project last year was also on machine learning. And I guess I can give a brief overview as well. The immune footprints that I talked about are antibodies which the body produces in response to whatever disease that it interacts with. And by looking at its protein amino acid sequence, which is basically like a bunch of letters that are put together, I was able to use machine learning to figure out from what diseases do these antibodies originally originate. And that allowed me to essentially deduce what the disease the person had based on their antibodies. So that's sort of the agent, and that essentially is a new disease detection method.
So that's really interesting. How exactly did your model work, and what kind of data did you work with?
Yeah, so I worked with, I guess, data from a lot of different sources, and the primarily source of my data, since I was working with these antibody sequences, was from, I guess, supplementary sections on data, on papers, sorry. And also there were certain databases that I looked into. So the NIH has something called GenBank, where it stores a bunch of different genomic related data. There was also a general antibody database for COVID antibodies specifically, by some group at Oxford. So it was from a lot of different sources. And I guess the model, what was special about it was that it was using sort of these large language modeling processes that we hear about more and more with ChatGPT and all sorts of these big generative AI models. And essentially what that did for me was it would take these antibody sequences and then convert them into a vector embedding representation of essentially what was just 1,024 numbers. And these decimal values, based off of that, I fed that into a feed-forward neural network, which gave me my final result. But I feel like this intermediate step of converting the sequences to a embedding representation was very important in terms of giving greater interpretability for the feed-forward neural network to build off of. If I had just tried to feed in the sequence directly, I don't think it would have been as accurate.
Yeah. Wait, so did you use like tokenization?
Yeah, so essentially how these, to my understanding, these LLMs, large language models, essentially work is it'll tokenize, right? With the English language, you have somewhere like hundreds of thousands of possible different tokens. Luckily for these amino acids, there's only 20 possible tokens. So there are 20 common amino acids. There are actually a few more rare ones, but we don't really care about them. These 20 common ones, in plain text form, they're represented by letters of the alphabet. So you'll see like A, B, C, D, E, F, G, so on, and arranged in different orders. And so those are sort of the tokens, I guess, that the model would use. But I think the nice thing about BERT specifically was the embedding representation model that I used is that it is context dependent. So based off of the position in the sequence, and I don't know if you've looked into this as well, the embedding representation will change. So let's take a word, for example, right? If we have like a pencil in the traditional embedding sense would just be, okay, we're going to have one specific embedding, one specific set of numbers for pencil, no matter what, no matter where it is in the sentence, anything about it, it's just going to be the same. So that sort of acts like a dictionary definition, and that is nice. But, the context will change the meaning of the word. Like, let's say, oh, can I borrow a pencil for a test versus let me pencil that in. Pencil in the second one is a verb. The first one is a noun. Obviously, they have different meanings, right? So if we have different embedding representations based off of its placeage in a sentence, then that'll be more useful. It'll provide more, it'll be more useful for us. So similar thing for these antibodies, because the position actually changes a lot about the overall protein, the overall antibody in itself. If you have sort of it at the beginning, that'll cause a protein to fold a certain way. If you have it in the middle and the end, it'll be different ways. So having that was very important. And there are other embedding models out there, but I think BERT is probably among the most stable that we have right now. So that's what I went with.
Yeah, I think it's like a really cool aspect of transformers that you mentioned there. And also, I one other question I had was, what kind of format was your data in? So you mentioned like it was genomic data and it was like sequences. So was it like raw text or?
Yeah, so a lot of it was similar to raw text. They have a special file called a FASTA or FASTQ file, but at its core, it's really just a bunch of lines in a text file. And so that's nice in terms of having the actual sequence. A lot of times, the data I was coming across they would actually interpret these sequences with different whatever physiochemical properties. So then you would get a bunch of numerical decimal values. And that I didn't really want to use because I wanted to really base it on the sequence itself. Because I feel like that's sort of the raw data, if you will. The other values that we get out of it are sort of algorithmically applied. We've interpreted our own stuff from it. So that's sort of a layer of human interpretation, which may or may not always be correct. The sequence itself is what we know is actually the sequence of the antibody. So yeah, it was in that sort of text format. There were some CSV files, Excel files where it was just down a column. And in Python, there are many different ways to work with that sort of data. But that was the general format, yeah. Yeah, definitely. So, I mean, if anyone's listening to this wants to get started with machine learning and AI, probably the most core is the language itself, Python, and you need some sort of IDE, integrated development environment, to actually code your Python in. So I just use VS Code. But I will say another thing that I found really useful was Jupyter Notebooks. Jupyter Notebooks are basically mini bits of Python files that can all be run separately. So you can write some code and then you can make a new block, is what it's called, of code. And you can run these blocks of code separately. So this makes it really easy to debug, especially is if you have an error at one block, you know that the blocks before were all fine, so you can just play with the code that was in a previous block and fix that up. I also use TensorFlow as my main machine learning framework. And I was trying to look into PyTorch. I would recommend PyTorch, I would say, is more advanced, but it does allow greater customization. And especially as you start to dive into transformers, which are all the big talk nowadays, learning PyTorch at some point is worth it. But at the beginner level, I wouldn't recommend it because I feel like it's just very overwhelming in a sense. What else besides that? NumPy as just for generally formatting numbers, pandas for dealing with these CSV files and just the initial import of the data. And then on the other side, once you're done with the machine learning, you have different analyses that you need to perform. So whether it's using matplotlib to plot graphs, or Seaborn can help with some graphs that matplotlib is not as good at. And there are, I guess, other miscellaneous libraries. There are some like bioinformatics Python libraries that I use to convert sequences, but that was more domain specific.
Yeah, that's all really cool. One thing unique about your STEM project that me and Armaan heard about at the start of the year when you presented it was that you got your work patented. So for our listeners who might be thinking about getting a patent or are wondering how that occurs within the field of artificial intelligence, could you describe more about that process as a whole and how AI played a role in it?
Yeah, certainly, definitely. Right. I think that's something that initially I sort of didn't know what exactly that was going to entail, but I thought about if I really have anyone listening to this, if you really do believe that you have something that's new, that's unique, that hasn't been done before, I would suggest just try it, just go for it. These sort of opportunities, you don't know what will end up coming down the road. But yeah, the patent process, what a patent does protect is sort of, it can protect a specific description of a process or a model, I guess. So the easier examples with physical models, if you made like a specific contraption or engineered some sort of device, like a I don't know, some sort of, let's say for a car, right? You engineer some sort of fancy exhaust system. You can patent that specific implementation of it. But the challenging thing with patents is that you also want to be broad enough that other people can't just change one thing and then bypass your patent. So they're sort of patent, a lot of the language, if you do read a patent, is sort of a bunch of like, this could be the implementation. They just like cover a lot of different options because they're just trying to cover as broad of a field as they can. And with software and artificial intelligence, it's a little bit different because you don't have a physical thing. And you sort of have to be, I guess, careful because sort of how patent law is built up is a bunch of different court cases that end up deciding different things. And then based on that precedent, you get into, it is the thing that you're proposing actually patentable. So for me, what I try to patent is really the specific process that I was using of going from the antibody back to the disease, and then using my specific machine learning model that I developed to be able to do that. And so what I ended up doing was not a full patent. It's called a provisional patent, which that basically gets your foot in the door for about a year. And yeah, you have 1 year if you choose to convert that to an actual what's called utility patent. And I'm in the process of figuring that out still. I haven't done that because it's a lot more expensive. But with the provisional patent, it's nice because you can sort of be a little more vague in terms of your description. So I just said, I'm using these amino acid sequences from antibodies, and I'm going to go back and I'm going to use that to figure out what is the original causal disease. And I've used this sort of neural network to be able to do that. So that's sort of what I wrote up. And yeah, the thing with a patent, I will say this, is that it's sort of a balance between trying to be broad enough but also not being too broad, because then your patent might probably get rejected. And specifically in the antibody domain case, there was a recent actually Supreme Court case where they said that, well, there was one company that tried to patent like a general format of antibodies, and then the Supreme Court said that you can't do that without actually describing how to do it. So you can't just go out and like patent an idea, essentially, or like a general framework. You have to patent a specific implementation of it. Yeah.
Yeah, that's pretty cool. I didn't know that you had to maintain that balance between vagueness and specificity within a patent. So that's interesting. What were the challenges that you faced with your project as a whole, or with the patent process, either one?
Yeah, certainly. I feel like there were a lot of challenges. So I'll share this with my personal story. How did I come up with this idea in the beginning? Because it seems like a very niche project in terms of a lot of detail that you need to get there. So I started working at a lab, before junior year, the summer before. But I wasn't really doing any biology there. I was doing much more just web design work, full stack work, and building a database to manage their patients that they were using for research or whatever. But they were dealing with a disease called leukemia. And leukemia is basically a blood cancer where it causes the cells that produce antibodies to overstimulate, to overproliferate, and then your entire blood just becomes antibodies. The technicians would tell me that they draw samples from these patients. Normally your blood is red, right? Their blood will be 90% white, which is kind of scary. And so that's sort of the problem, the extent to which it gets. But the thing with that, right, is then you're going to have antibody patterns in these patients because it's just all clonal. It's all like a tumor that just like expands throughout the body. But you're going to have specific types of antibodies that are common in these leukemia patients. So that was a fact that I sort of learned about, but I didn't really pick up on during the summer. But when it came time to do my STEM project, I revisited that and I thought about, okay, that's pretty interesting. You know, can I apply what I've previously learned about machine learning to this and maybe just start with using that in differentiating leukemia patients. CLL is the type of leukemia from healthy control patients. Mm-hmm. So that was just my two categories that I wanted to do. And I will say the biggest challenge there was actually finding enough data for the leukemia patients. It was very hard to find the data that I wanted in terms of the antibody sequences from patients who just had the disease and to find a lot of it because there really was no centralized database for it. So I tried that for a little while, and then this was kind of just on a whim because I was like, things aren't really working. Can I try this for another disease? And it was just a question. I really didn't know the answer. And I just happened to stumble across the COVID database that it was like 10,000 different antibodies. So I was like, wow, okay, that could work. And sort of the big aha moment there was that this analysis isn't just restricted to the specific case of leukemia where these patients have clonal antibodies. If I am correct and my sort of hypothesis works, that I can use this to diagnose or detect diseases, then it's applicable for any disease basically with an immune response. And that just opens up the option so much more. Mm-hmm. So I went with COVID, and then I was the flu and HIV and a few other diseases as well. So I was able to do a lot of diseases now instead of just being restricted to leukemia. And so that opened me up to a lot more data as well. And going from there, I think, was really just the coding process. And the coding process is never going to be smooth sailing. It's going to have bugs, and it's going to involve a lot of searching up error messages on Google and that like. But I feel like the thing with coding is you get better as you do it. Even if it's really slow, if it seems like it's really slow, you are getting better, and you are getting more used to the system. And eventually you'll be able to solve issues pretty quickly. Mm-hmm. So that's sort of what I found. And I will say that there's sort of a bad reputation of, coding is all just sort of copy paste. And yes, but after you've written the initial code. So writing the initial code is the hardest spot. And then you're able to copy paste a lot of it. But you also have to be careful while you're copy pasting it, because if you accidentally copy paste something and it's not correct, then that can completely ruin your entire code. So I'll share one example is I assigned like data for COVID to one variable in Python, and then data for the healthy patients to another variable. Mm-hmm. And I somehow copied it. And I thought that I had kept the two variables separate, but somehow it was called COVID sequences and then healthy sequences. And somehow it just ended up being COVID sequences. So I was only using COVID sequences while I was trying to differentiate between them, because my labels were saying, half of these are COVID, half of these are healthy. And so you can imagine that the model was stuck at 50%, which is just random guessing. For a while, I couldn't figure out what was going on for the life of me. Mm-hmm. And I had to carefully reread the code. And eventually I saw it and I'm like, okay, that's the simplest mistake ever. But once you're able to solve it, it is nice and you can move on. Yeah.
Yeah, no, I totally see what you mean. Like on a project I was working on just like a couple of weeks back, I just like labeled the training and test data, or the training and validation data, with the two wrong variables, and they were like completely switched around when testing for accuracy, and it was so annoying until I finally found that bug. But yeah, moving on to the next question. If you could do your project over again, what would you do?
Ooh, good question. If I could do it over again, I think I would start with the realization, that this isn't just leukemia, because I spent I stayed with leukemia for like until like mid-October. So that was like a month and a half that I think I could have branched out to more diseases. And I think that the biggest thing, I think I would have done things pretty similarly. It's just sort of maybe I would have had been able to expand my scope and how much sort of the scope of the work. And I think that, yeah, there are a lot of things I guess I didn't have time to try within just a time frame. Like there are these antibodies, but then there are also what are called T cell receptors, which are the other main type of immune receptors that you see in disease responses. And so those ones are more evident in cancer. And if I was able to expand to that, I think I would have had some interesting results in cancer, as well as just mainly I was working with infectious diseases with these antibodies. I think the other thing was just, yeah, a lot of the machine learning ended up being limited by how much computing power I had, because these models would take like hours upon hours to run. And yeah, I think that was another big limiting factor, just the amount of computing power that I had, and how far could I go with that. So generally, I think you asked, yeah, what would I do if I could do it over again? I think things would be similar, but I would branch out a lot more, and yeah, hopefully that would be more interesting. Yes, I did train locally. So I would say if you're going to start with a very small model, you can try a Google Colab. Google Colab is on the browser in the cloud. And it's much easier because you don't have to deal with the hassle of the setup. I have a M1, right? And an M1 is probably the hardest device to set up TensorFlow and all of these libraries on, because the whole thing with like Apple Silicon and Intel incompatibility. But you sort of Google Colab, I'll be honest, its computing power is kind of trash. Like once you get past a certain point, it really can't do much, and it's like taking 12 hours to run a model, which is longer than it actually lets you stay on Colab for. So yeah, I had to move to
This is why you
local
buy Colab
and
Pro Plus, by the way.
Colab Pro Plus, so that's like $50, right?
Yeah, I just bought like
but
the normal like Colab Plus today, just like one stem thing.
Yeah. I mean, yeah, look at if that's worth it for you, because otherwise it's investing in a large piece of computer hardware, which, you know, luckily I did have already. But if you don't have that, that might be worth it if you're just using it for a few months. But yeah, that aside, I mainly stuck with, I guess, my computer because it was fine. Like, yes, there was more that I wanted to do, but things weren't outrageous in terms of runtime, and it was still able to do things well. And you know, I know WPI also has servers that if you want access to, and their servers are just crazy powerful, but it's a lot more of a learning curve, right? So it's a trade-off of, do you want to spend the time learning how to do that? And then I think debugging there is also harder because you can't see the outputs from Jupyter notebooks. That's another thing that I think I've come to rely on a lot, because yeah, like sort of having that feedback as I go is very important, rather than just running one file at the end and having to add a bunch of print statements everywhere to see where the error is. So yeah, I did things sort of locally for now, but I know that obviously if you're going to really expand to larger models, if you're going to train your own LLM, large language model, yeah, look into servers for that point, because you're not going to be able to buy, like, if you want to set it up at home, it'll cost like easily $100,000, and yeah, that's just crazy.
Yeah. Yo, I'm not gonna, like, my STEM project actually is training an LLM, and I don't have like access to the WPI servers. So I got like something like decent going with like GP2 checkpoints and like Colab. But like, yeah, it's kind of crazy if you have like larger scale data sets and you need like an insane amount of VRAM as well.
Yeah, RAM is the other big limiting factor. So I will say, like, for anyone who wants to get started with machine learning and wants to actually like invest in computer hardware, get a good GPU, but more importantly, get a lot of RAM, because everything ends up getting loaded into RAM. And so I have a Mac, right? I think Macs are nice because you have the unified RAM, and sort of that allows you to get much more RAM than like NVIDIA GPUs, where I'm pretty sure the largest one normally is like 24 gigabytes, which is good, but even some models won't fit in that. So, yeah.
Yeah. All right. So what's our last? Okay. Yeah. I guess like one last question we had was like, are there any
Sure.
other AI projects that you were working on, or anything you wanted to share about?
Yeah, sure. So, other AI projects, I guess I've taken a little bit of a break. I've started to work on this project in this sort of disease detection project in a larger capacity, in terms of also trying to branch out to um, cancer, cancer detection. And I'm trying a different approach there in terms of more clustering of these embeddings from these large language models, because the thing is, the limitation of neural networks is that you have to have output classes at the end of it. So you're kind of restricting yourself to which are the possible output options, and if you get data that doesn't fall into one of them, it is like guaranteed to make a wrong prediction, right? So I think clustering is a lot more computationally expensive, but it's worth it because you are not limited to having to define your own artificially constructed clusters. Artificially constructed neurons, or like output classes. You can see what clusters are naturally there and then sort of work with that.
So
And
it's like an unsupervised learning approach.
yeah, more like unsupervised learning. I feel like unsupervised learning is, at the end of it, is once you dive into higher level ML, that's sort of where things go in terms of, and I'm pretty sure LLMs sort of are like that. There's like self-supervised learning, which is a variant, but yeah. Other machine learning projects, if I think really quickly, let me think. Sort of, I wouldn't say there's other big projects, because I feel like big projects do take up a lot of effort, and but if it is worth it if you want to do it. Sort of having the skill though of being able to do ML is really useful. Like I will say, in math modeling class last year, like there were several projects where I could just sort of use machine learning to find the most optimal answer, because at the end of the day, machine learning is just math, and it's really complicated math, but it's like optimization math. And so having that skill of being able to know, like, okay, what sort of model would best fit this data, and then how do I craft a model like quickly. It doesn't have to be super complex. It's just something that gets the job done. That's sort of just like having more of the skill rather than the big project. Yeah.
Yeah, absolutely. Thanks so much for the interview, Joseph. We'd like to ask if you have anything else you'd like to leave off our listeners with, regardless if it be about AI or life in general.
Yeah, sure, definitely. And this was great. Thank you for having me. I think that generally speaking, what I've learned, I don't know if this is for a Mass Academy audience or just in general, but sort of take advantage of the opportunities that you have. I think that's what I really did in terms of WPI, and taking advantage of all of the access and the resources that I had there, because it started with the patent in terms of getting that through with them, but then also I've been able to explore more into their business school and then things like that, and then different pitch competitions, and I've actually been invited to a few because of that. And none of that would have happened if it does start with like one step and you kind of don't know where it'll take you. But I would say just wherever you go, it's sort of, however much you get out of whatever setting that you're in is based off of how much you take advantage of the resources and the opportunities that you have. So I guess that would be my major suggestion, and that sort of is what made, Mass Academy and everything really worth it for me, more than, academics are wonderful, people are wonderful, but it's really the resources, having access to university resources as a high schooler. There's just so much that you can do. And so I would encourage everyone to explore.
Yeah, that's really great advice. Just to conclude, thank you to all our listeners, thank you to Joseph, and make sure to tune in next week for our new episode. Armaan, Vasu, and Joseph out.